Dr. Erica Hartmann from Northwestern University, who will be giving us a talk on using Pangenomics to monitor the built environment.
Great, thank you. And let's see, this works. Oh yeah, look at that.
Got it.
All right. Well, I apologize for being what stands between us and lunch, but somebody has to do it. I am going to talk a little bit about pangenomics to monitor built environments, but I'm actually going to start out with a little bit more about some of the other work that I do that I think could also be very relevant to this crowd. And I'm going to start out by, so I've been told that there are some non-microbiologists in the environment, in the audience. And so I thought I would start by defining, at least from my perspective, what a microbe is. And this is somewhat modified, I believe, from Bruce Rittman, who says that microbes are bags of enzymes. I would like to modify that to say that they also respond to physical and chemical stimuli, and we've seen some of that already, especially with the presence of water. So obviously also, we know that there is no such thing as a sterile built environment, and especially we as hosts are bringing our microbes in with us everywhere that we go. Especially, I think, in the context of the International Space Station and these sort of high-risk environments, also with hospitals, we're very concerned about the fact that we are bringing these microbes with us. And so we do all kinds of things to sterilize or sanitize our built environments and to prevent the spread of pathogens, as Kath just discussed. And one of those important things is hand hygiene. Kath alluded to this briefly, but it turns out that most people on average wash their hands for about 7 seconds. And I will say right here now that that is insufficient. So if you really want to wash your hands properly, I think it's supposed to be for 30 seconds, so you're supposed to sing the ABCs twice. So that's my public service announcement for the day. So we do all of these things to exactly prevent the spread of pathogens or unwanted microbes, maybe the things that are going to be responsible for biocorrosion or what have you. But in addition to doing things like practicing proper hygiene, we also introduce all kinds of chemical barriers into our environments. And this is one of the things that I think is really, really interesting about engineered environments is that we can control all of these different chemical exposures. But I think right now we don't do so consciously. Hopefully you'll see by the end of my talk that we could think about it a little bit better. So some of these chemicals include things like Triclosan in personal care products, Benzalkonium chloride, which is now in antibacterial hand soap, and as well as, of course, all of the prescription medical chemicals that we use. And I think also I really like Kath's demonstrations of mixings within built environments, and we've already heard a little bit about chemical emissions from within built environments. So to really think and to really drive this home that built environments are not just sterile, static systems, despite what the astronauts may say, that they really are dynamic chemical and biological systems where maybe you don't necessarily have so many outdoor sources in the International Space Station, but we have all of these different sources of chemicals, both the ones that we introduce intentionally and the ones that sort of get in there unintentionally. And of course, all of these chemicals are going to encounter at some point or another the indoor microbiomes, all of these microbes that are, again, there either intentionally or unintentionally. And so to really think about how these microbe-chemical interactions, basically what they are and how we can either take advantage of them or limit them. So one of the interesting things about using antimicrobials to stop or prohibit the spread of microbes is that it almost inevitably leads to some sort of resistance. And so basically anytime a microbe encounters some sort of barrier in some period of time, whatever that is, it will find a way to get around that. Like I have complete confidence in the microbes that they will always win. And they can do this in a variety of different ways. Some of the interesting ones are through transformation here, so actually with a lot of organics, finding a way to actually use that antimicrobial supposedly as a source of carbon and energy, or through efflux, and this is one of the things that is very concerning in terms of the medical field because some of these efflux pumps are non-specific, and so you can develop cross-resistance between different antibiotics and antimicrobials. And antibiotic resistance obviously is very scary on the International Space Station because, you know, what would you do about it? But it's also just a really big deal worldwide. So there are currently about 300,000 deaths, there are currently about 25,000 deaths, I believe, in the US and in Europe that are attributable to antimicrobial resistant infections, and that those numbers are expected to increase by 2050. So basically, I would say anything that we can do in any capacity to limit the spread of antibiotic resistance is very important. And the good news, I would say, if there is any, is that at least there are a lot of people who are paying attention to this, and so hopefully we'll come up with some good solutions in the not so distant future. So for the purpose of this talk, and just here, just now, I know this is not standard nomenclature, I'm going to differentiate between antibiotics and antimicrobials, and I'm doing this just from a functional perspective of like the way we think about why and how we use these chemicals. So when I say antibiotics, what I really mean is medicines for humans or animals. So these are going to be the things that you would take if you had an infection. And in contrast, antimicrobials are the sort of products that we surround ourselves with. They're not necessarily used for medical reasons, and so the example I'm using here is antibacterial hand soap, but this could be any number of products. We already heard about silver-enabled surfaces to prevent biofilm growth. So all kinds of products contain antimicrobials. And the big functional difference that I want to emphasize, in addition to the whole prescription use versus over-the-counter kind of thing, is this idea that antibiotics are given typically for short periods of time at very high doses, whereas antimicrobials, these are things that are going to leave long-lasting residues or be present for a long period of time. And that's important if you're a microbe because the longer you're exposed to something, the more chance you have to evolve some sort of response to it. So I'm going to tell two different stories about antimicrobials before I get into the pangenomics work. The first story is about Triclosan. This is an antimicrobial that is currently used in toothpaste and other goods. It is incidentally ubiquitous in indoor dust because it's such a high production volume chemical and was used so widely for so long. It's basically been found in every dust sample that's ever been sampled worldwide. Exposure to Triclosan for microbes has been linked to antibiotic efflux, so these are these multi-drug efflux pumps. And it's also linked to horizontal transfer of mobile antibiotic resistance genes, and this will come back a little bit later, but basically the idea that antibiotic resistance can travel between microbes makes it that much harder to control. So we wanted to sort of probe this relationship between Triclosan in dust and antibiotic resistance, specifically in the dust microbial communities. And so to do this, we collected a bunch of dust samples. We ran around with vacuums. And from that vacuumed dust, we measured and extracted Triclosan using liquid chromatography tandem mass spectrometry. So this is a good way to get positive identification and quantification of a specific chemical. We heard a little bit about using untargeted metabolomics yesterday. I think it would also be interesting to use a sort of untargeted approach in maybe future analyses. But anyway, for the purpose of this study, we just wanted to know about Triclosan. We also, from these same samples, extracted DNA, which allowed us to identify antibiotic resistance genes from the dust metagenomes. And then we went super old school and we also cultured antibiotic-resistant bacteria, and we specifically screened for resistance to 3 different classes of antibiotics. So this is work that was led by Ashkan Fahimipur, who at the time was at the BioBE Center at the University of Oregon. And basically what we found is that some some of the microbes that were enriched in dust samples that had higher triclosan concentrations are microbes that have more resistant genotypes. So those taxa typically have more antibiotic resistance modules within their pangenomes. So we're seeing something about samples where we have more triclosan. We see bugs that are typically associated with more antibiotic resistance, and this is from the metagenomics data. Metagenomics is great, but we've already heard about not all sequencing can confirm viability, much less phenotype, and so to back it up, we have this now culture data, and we're still playing with all these. I think we have, I think it's 2,000 or 3,000 isolates from these samples, so it's quite a few isolates that we're playing with. But for the first pass, what we can say is the density, so the number of colony-forming units that we got per milligram of dust, as well as resistance to these 3 antibiotics, clarithromycin, ampicillin, and tetracycline. And I just want to highlight this cluster here. These were buildings that had higher triclosan concentrations. They sort of clustered together. And here we also see that they have a higher tendency to have multiple resistances to multiples of these antibiotics. So we still don't entirely understand what exactly the relationship is between triclosan in dust and antibiotic resistance in dust, but certainly there's something there. So for those of you who have been paying attention, if you're really concerned about antibacterial hand soap, you will know that the US FDA recently banned triclosan along with 18 other antimicrobials as the active ingredient of hand soap. However, you can still buy antibacterial hand soap. I'm not sure why, because no one's ever shown that antibacterial hand soap is any more effective than regular hand soap. So my personal advice to you would be to go out and get regular hand soap and wash your hands for 30 seconds. But anyway, you can still buy antibacterial hand soap, only now instead of triclosan, it will contain benzalkonium chloride. One of the things that's interesting about benzalkonium chloride is that it is known to be ineffective against spores. And spore formation is one way that microorganisms can be very resistant, not just to benzalkonium chloride, but all kinds of environmental stressors. In addition, exposure to benzalkonium chloride has also been linked to antibiotic transformation and efflux. So again, continuing this relationship between antimicrobial exposure and antibiotic resistance. So in addition to being in antibacterial hand soaps, benzalkonium chloride is also the active ingredient in the world's first microbicidal paint. And so we did a little study to look at what's going on there with these antimicrobial-enabled surface finishes. So paints often have an antimicrobial added to them. I would just like to point out that the reason that we put antimicrobials in paint is not to protect the people in the room, it is to protect the paint in the can. So if you leave paint for a really long time, it will grow mold in the can. So that's why it's there. That's not necessarily clear from the advertising, but that's another story for another day. So we got a few paints and we decided to look at whether surfaces that were painted with these different paints were in fact effective at preventing microbial growth. And this is work that was performed by my Ph.D. student, Jinglin Hu. And so we have this microbicidal paint that I mentioned has benzalkonium chloride in it. We also had a conventional mold-resistant latex paint. And just for good measure, we threw in a clay paint, which is, people who are thinking about sustainability and low carbon footprints sometimes are using these more natural building materials. And so we wanted to have that in there for comparison. So the first thing that we saw is, as you sort of might expect, we didn't see growth, nor were we expecting growth. We are basically looking at decay and then what survives despite that decay. And in general, bacteria do not like being on dry, barren surfaces. So regardless of which paint we used, we saw a decrease in the number of viable bacteria over time. But what was interesting is that depending on the surface, we would see survival of different members of our microbial community. And I should have said our microbial community was composed of isolates that we collected in the previous study. So these are authentic built environment dust undomesticated isolates. A lot of times when we test the efficacy of antimicrobials, we use, ATCC strain, blah, 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 that has been kept in the freezer for 90 million years and is incredibly well studied, but may not in fact be representative of what would be encountering these things in the real world. And so one of the things that we wanted to do was to have this sort of step towards a little bit more realism. So we saw that we looked at 3 different relative humidity settings, basically. So as we already heard, 100% relative humidity is basically what you need if you're going to have any sort of growth. We didn't see growth, but hypothetically, that's what you would need to support growth. And then we also had ambient, which in our lab ended up being about 20% relative humidity. And then we threw some desiccant in to get something really, really dry. And we can see that depending on whether you're looking at, in this case, the clay paint or the conventional paint, you would see enrichment of different members of the community over time. So again, these are all dying, but they're dying basically at different rates. And if we looked at the antimicrobial, the microbicidal paint, the thing that survived the best was this Bacillus timonensis. And again, if you're paying attention, Bacillus is a known spore former, and benzalkonium chloride is ineffective against spores, so maybe there's something going on there, maybe sporulation is involved. So we did some microscopy to look at that, and so basically this is our Bacillus timonensis authentic undomesticated dust isolate, and this is it in vegetative state and the spores. And these are from swabs that we collected from the surfaces. It's a little difficult to see, but you can see both the vegetative and the spore forms. So certainly sporulation could be part of what is keeping these cells alive, but it doesn't explain the whole picture. And so we'll be following that up to sort of see if we can identify other resistance mechanisms or whatever else is going on. All right, so I promised I would talk about pangenomics and the International Space Station, so here we are. I think part of what came out of our previous work was that basically, yes, exposure to these different chemicals is important, especially for looking at the different phenotypes that they might induce and with regard to antibiotic resistance, but there are certain stressors that are just common to the built environment. Just the fact of being on a dry, barren surface will change, obviously, the behavior of a bacterium versus being, in culture media. And so originally what we wanted to do was to do some sort of comparison of bacteria that had been isolated from surfaces within the built environment and bacteria that had been isolated from, in this case, we were looking at either soil-associated bacteria, so isolated from the soil, or human-associated bacteria isolated from, for example, the gut or the skin. But what we found is that there were not enough isolates from the built environment to do as big of a comparison as we wanted. However, there was one built environment that is incredibly well studied and had this wealth of isolates that have been collected over the last 20 years, and that was the International Space Station. So I just really want to say thank you to everyone who has been collecting these isolates for this period of time. I think this is a wonderful resource for the microbiology community. Thank you especially to Sarah, the genomes of whose isolates we looked at in this study. I know that molecular biology is the future and everybody's very excited about all the things that we can do with sequencing. I just am so happy that we also have isolates to play with because I think that's so important. So we have these isolates that have been collected from surfaces within the International Space Station, and we can compare those to similar isolates or similar taxa that were isolated on Earth, either associated with natural environments like soil or humans. And we looked at the pangenomes of these organisms. So this is this idea, for those of you who are not familiar with pangenomes, just really briefly, that microbes have an enormous amount of genomic plasticity depending on the different taxon. So each group will share a core genome, the amount of genes, certain genes that are shared between all members of that group, but then there will also be these accessory genes. So when I was talking about, for example, horizontal transfer of antibiotic resistance genes, those would fall into the accessory category of things that not all members of that group have. And so you can compare if members of a group in a certain context all have genes of a certain function, and you might infer then that that function is important for survival in that context. Okay, so that's what we did, and basically we wanted to try to answer some of the questions that were raised earlier, that Rachel talked about, this idea of, do microbes actually adapt to the built environment? Is this evolution actually occurring? And so we picked for this 2 model organisms, Bacillus cereus and Staphylococcus aureus. We picked them because they were prevalent within the ISS culture collection. I promise NASA every time I give this talk, I do clarify that the space station isn't just like coated with staph, it's just that those isolates were most prevalent in the collection of isolates that have been retained, right? So I try to be very clear about that. These organisms are also interesting because they differ in lifestyle, survival strategies, and virulence. So I already mentioned Bacillus is well known as a spore former. Staph more commonly will form biofilms, so we thought maybe these differences in lifestyle might contribute to different adaptation strategies to these built environment contexts. And this was work that was performed by my postdoc, Ryan Blaustein, again with the fabulous support of Sarah, who's in the audience. And what we found is that both of the pangenomes of these 2 organisms are open. So basically, every time we get a new genome of one of these species, we are adding new genes. But the rate at which we are adding new genes is different for these 2 organisms. So we are adding more genes per genome for Bacillus cereus than we are for Staph aureus. We think this might have something to do with the fact that A, Bacillus cereus has a larger genome in general, but also B, it's more of a generalist, and so maybe there are more different environmental niches that it's occupying as opposed to Staph aureus, which is basically going to be on human skin more or less, plus or minus. But we're not really sure, and that would be an interesting thing to continue to think about. We can also see that the relative size of the core and the accessory genomes for these 2 organisms is different. So Bacillus has basically a very small core genome and a very, very large accessory genome, whereas Staph, about 30% of the genes within that genome are core, so shared between all members of that species. This is Staph aureus. And when we looked at the genes that were specifically enriched in the accessory genomes of these 2 organisms on the ISS versus either in soil or associated with humans. I don't expect you to like read this whole heat map or whatever. But what was interesting is that the genes that were enriched tended to do with biosynthetic pathways, stress response, and energy transport. And so the way we interpret this is that basically both of these organisms have gone from where they were happy and healthy to these dry, barren surfaces and are having to really scavenge. So possibly they're responding to changes in temperature or moisture availability. Possibly they're looking for new carbon sources or other nutrients. So basically all of these things are things that one might project would be useful to microbes that are all of a sudden in this hostile built environment. What's reassuring is that none of these things are associated with virulence or antibiotic resistance per se, and so we don't have any reason to assume that these organisms are becoming more pathogenic in the ISS. We can't say that that's necessarily the case for all organisms. Of course, we think different microbes will respond differently. I don't think there is, and as has already been said, a one-size-fits-all model for this is what happens to microbes in space. But at least for these 2, we can say that with the evidence that we have, they're probably not that big of a risk, which is very reassuring. And I also sort of wanted to go back then to this idea of viability and horizontal gene transfer to say that while obviously to be actively adapting and evolving, we need things that are viable and metabolically active. However, there's potentially still a role for dead bacteria and dead organisms in this whole process, and that could be as a source of genetic material for other viable organisms. So within the built environment, we have all these different sources of organisms. We have the occupants and of course the dogs, which are very important. We also, maybe less so in the International Space Station, but in other built environments, we do still have some influx of organisms from outdoor air, outdoor sources. And then of course there's the community that's actually just there, maybe growing on the building materials. Who knows how long they've been there. And so these two, the outdoor and the indoor sources, can deposit into indoor dust. And then the indoor dust can be resuspended, as we just saw, which can serve as a new source of exposure for the human occupants. But in addition to that, there can be genetic exchange between organisms in all of these different groups. And so wherever things are coming together, it's possible that the genes could be taken up. And there's increasing evidence that this is possible, that you can actually have transduction from a dead cell into a live cell. So I think this will be a really interesting area to continue to look at how, basically what's mediating those transfers and what we can do in terms of selecting chemicals or designing airflows to control those kinds of processes. So at the beginning of my talk, I defined microbes as bags of enzymes that respond to physical and chemical stimuli. But particularly within the built environment, we are engineering those physical and chemical stimuli. And so I think it's interesting to be able to ask this question of specifically how do these different stimuli affect which microbes are there and also what they're doing. And how can we leverage those stimuli and our ability to control them to control basically the microbial processes that are happening all around us. So with that, I invite everyone to think not just about within the International Space Station, but within the environments that are around us all right now. How do your choices about the kinds of materials that you're using affect the things that you are exposed to? So I'll just let everybody marinate on that. And with that, I'd like to thank all of my collaborators, all of our funding sources, my fabulous students, and I'll take any questions.